All right, so I think we are good to begin with today's session. I would like to welcome you all for the first session of getting started with bioinformatics in R. So let's quickly discuss that why this program and what are we going to discuss today. So as biology is saturated with complex data sets that have to be sorted, explored, and looked into, anyone handling data generation, analysis, or decision making based on data has to gain some level of data science skills. And the increasing necessity to process big data and develop algorithms in all fields of science means that programming is becoming an essential skill for scientists, with Python, the language, and also the R of choice for the majority of bioinformaticians. In most biological and biomedical settings, you will be expected to run or implement programs written in R and Python scripting and others. So, getting started can be hard. Programming is like learning a new language altogether. And that is why we offer easy to follow, structured, and simple coding tutorials designed around bioinformatics challenges. So, without any delay, let's get started with today's session. But before we explain what we're going to cover and also get started with the session, I would quickly like to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools. And our mission is to make bioinformatics more accessible. The Omics Logic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. Over the past several years, we have had the privilege of working with a number of organizations developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. Omics Logic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. I would like to take the honor to also introduce the founders and team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of inflammation. And one of his major projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. And our platform is a result of hard work by some of the people who are mentioned here on the right. Julia Panov is an expert bioinformatician. Avi Titovsky leads the development of the T-Bioinfo platform. Dr. Mohit is an experienced bioinformatician with multiple publications in peer-reviewed journals. And Dr. Raghavendran is an accomplished computational biologist with an experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. And my name is Sonalika Ray, and I'm the Omics Logic Community Manager, and I'm responsible for daily interaction with thousands of users that are a part of a community managed by Pine Biotech. So now let's quickly gear up for today's session. So the very first important thing that you all need to do is to make sure that you have an account on the Omics Logic Learn portal, because today we are going to take you through the resources on the portal. We'll be practicing the sessions as well in the console on the portal itself. So it's very important for all of you to make sure that you have an account. I'll quickly give a demo to those who have joined the session and have not created an account yet. So this is the link to the portal. It's in the chat box. Everyone, yes. And I'll just give a quick demo how you can sign up if you have not done yet. And those who have signed up, please let me know in the chat box so that I know we are good to begin. All right, so you need to click on the login button, and then you can either sign up by creating an account for yourself, putting in the details required, or simply use any one of your social media accounts to have an account on the portal. It's absolutely free of cost. Now, I would be doing it with one of my Google accounts. You can create an account right away so that if you face any technical issue, or I'm here to help you out with it. And also I'm awaiting responses in the chat box from the people who have already signed up. Let's wait for a minute or so that we all have an account and we can follow the resources along the mentor. Perfect, Christine. Thank you for the confirmation. What about others? All right, so I'm hoping that you have signed up, or if not, then you have the link in the chat box. Please do so. The next thing is, after you have created your account, please make sure that you have updated your profile. You need to make sure that you have a profile picture, connect your social media links, and also add a brief bio about yourself. So today, the first session, I have invited those who had a keen interest to be a part of the program, but have not yet enrolled, right? So those who have enrolled, make sure that you have a starter subscription active that is going to give you the access to all the starred lessons on the portal. And also make sure that you have a badge of getting started with R on your profile. If you do not have a badge, or if you do not have a starter subscription button here, please let me know and I'll activate the same for you. The next thing is that having a complete profile is very important, because we are going to monitor your progress via your profile. So whichever step you are, whichever lesson you complete, we'll get a notification about your achievement via your profile itself. And once you have completed a particular course on the portal related to the program, you will be certified for the very same program. Here in the profile tabs itself, go to the certificate section. And once you have the particular course completed, you will get a certificate here. You can also look at your certificates for various coursework that you complete along each and every session of the program. And also on completion of the program, you're going to get your program certificate here under the programs section. So that was it about certification and profile. Now, the next important thing before we get started is to actually see how to navigate on the portal. So when you click on the courses tab, the very first thing, as a part of the program, you're going to have the access to all of the courses on the portal on multi-omics data analysis and data science for R and Python for bioinformatics. Each and every course you have an access to, and also the example projects. Now, how do you navigate? Either you simply write R. So because we have the R session at present, we'll be having the Python session after an R for the ones who registered for Python. So you need to go through the R lessons, specifically R coding course 1 is here and R coding course 2 is here. Now, the very first coursework, our course 1 is an introductory sort of coursework which will help you just get started with R. And in the second coursework, you'll be able to work with some statistical analysis and also learn about various visualizations, and definitely practice it alongside and have one for your own data set. All right. This is what I wanted to show about our lessons. Also, it's very simple to look at all of the courses that are there on the portal. Just click on the course tag that is there on the right side. All of the courses will come up here. And also, if you want to learn how after getting the like after going through a particular coursework, where in which particular fields can you apply the knowledge that you gain, or how you can answer a particular research question applying the knowledge that you gain from a coursework. We have various example projects. Now to explore the example projects, click on the project tab, and then you'll have various example projects. And we have classified it over here. So if you click on data science, you'll be able to just shortlist the exact project that you would be interested to go through to understand the application of the coursework. So that was it about navigation on the portal for various courses. And also now quickly, let's have a look on the program page. Now, those who have successfully enrolled for the program, they should have the badge of the program. Now, having the badge of the program on your profile will make sure that you are a part of this program page. Now, what do I mean by being a part of this page? If you scroll down, you can see that we have mentioned the details for each and every session here. The schedule for each and every session has been mentioned here. Now, as we have the very first session today, just after this session, after a few hours or so, you can see the recording of the session here on the program page itself. You can go through the recording once again to revisit the topics that we are going to discuss today. Also, along with each session, you can see the topics to be covered in the live session that we'll go through. That's what we understand. And also we have some coding lessons that you have to go through and complete. Now, these coding lessons are actually the lessons of the courses that I just showed you, our course 1 and our course 2. So we have structured it in such a manner that the mentor will be taking you through the topics, and then you need to practice it yourself from these lessons, right? Now, once you go through these lessons, complete all the assignments, and also you go through the recording, you need to click on the complete session icon. This complete session icon will get activated once you have completed the lessons and also gone through the recording. You need to click on the complete session icon and do so for each and every session on or after the session date. Now, by clicking on the complete session icon, you'll be able to complete all these sessions on the program page, and you will get your certificate again in the profile section. So that were a few important things that I wanted to discuss with the participants before we actually get started. Now I'll hand over the stage to Dr. Raghav. Let's get started with today's session. And today we'll be discussing about, we'll just get started first of all with R. We'll discuss about big data and bioinformatics, and also we'll learn to use the code playground, the inbuilt console on the Omix Logic Learn portal, and learn how to load data in R. Also, we recommend you all to please go through the very first coursework on the portal, that is Introduction to Bioinformatics. For those who come from a non-bioinformatics background, this particular coursework will help you get started with bioinformatics as a field, and also help you understand why bioinformatics is important, like what is the role of the field, and how does it help in translational healthcare, like what is its role in translational healthcare. It's a very short coursework and you'll be able to just complete it within few hours. So that is also one of your assignments for today. All right, so that was it. Now, if you have any of the issues, or if you face any technical glitch, then you can please let me know. I'm here in the session itself. You can let me know in the chat box, maybe or after the session, and I'll be happy to help you. Over to you, Dr. Raghav.
Thank you. Thank you very much, Sonalika. I mean, let's get started. Before sharing my screen, let me ask you, how many of you have had any sort of introduction to R before? I mean, not that it is mandatory that you should have, but if you have had any sort of introduction to R or using R, then give a yes or one in the chat box. So let's find a way to share my screen. Okay, we have some few inputs coming. That's really good. Good. It's not a prerequisite, just to get an idea of how many of you have had some introduction to R, and how many of you have had none, so that I can plan how deep I should go and from where should I start. Okay, thank you very much. I think I get a general idea of the distribution. Thank you very much. Keep coming. So hello and welcome everybody for the first session of the program, getting started with bioinformatics in R. Today we will learn about what constitutes a big data in the field of bioinformatics, and we will learn about why a programming environment like R is best suited for studying this big data analysis. And finally, we will get an introduction to R data types and how omics data can be loaded into this environment for further data exploration. So as we start, let's begin with what is bioinformatics. So if you are new to bioinformatics, as Sonalika mentioned, go through the introduction to bioinformatics course. That is going to help you with a lot of necessary knowledge about different topics that we are going to deal with, that we are going to discuss with today and also throughout this program. So it is a convergence of biology and data where both are equally important, as we will see in next few slides and also as we will see throughout this session. So who uses bioinformatics and for whom is it for? The answer might have been something like 10 years ago. The answer might have been for statisticians, computer scientists, or engineers. But today, the truth is that more and more biologists and clinicians are expected to understand, handle, and then analyze their own data to succeed or to publish in basic and translational research. So in fact, because of this change, the biology itself is rapidly changing and it's acquiring the character of data science. And what do I mean by that? So biology in today's era is actually can be analogous to data science of biological data. So where billions of data points on genes, proteins, and other molecules are compiled in large files and systematically analyzed or studied. So analyzing such data will lead to more knowledge and will lead to more understanding about the living organisms, including human living organisms, that will help us understand human health in different biotechnological areas, and it will help us understand crops, and it will help us understand areas like metagenomics and viruses, infectious diseases, and etc. etc. So this influx of huge volume of data and the development of associated methodologies has completely changed the biological research towards a data-driven approach for research and asking questions. So this change has made bioinformatics for life sciences as prominent as computers for tech industries. So we spoke about big data and their effect in changing the face of biology and biological research. So where is this data coming from? This field, the whole omics field, is based on high throughput technologies that will generate billions and billions of data points on genes, proteins, and other molecules that characterize biological system and phenomena. We will have molecules that characterize the phenomics of the cell, the genomics of the cell, and we will have epigenetic modifications, and we can also have transcriptomics data. So these data are compiled into repositories, and these repositories are available for many, for general public to download and analyze, so that they can be systematically studied to understand, interrogate, and to establish trends for many different applications, not only limited to biology, not only limited to clinical sciences, but many different applications in general. So as I mentioned, this is open for public. So is such data available? Yes, it is available. And one such repository is maintained by the National Center of Biotechnology Information, which has at its base a collection of peer-reviewed articles, biological data sets, and projects on various different topics and various different data types. As you can see, in the past several decades, this resource has been growing exponentially, and the data that is submitted has been so rich, so that the analysis methods and analyzed data has not been able to catch up. So to give you a perspective, there are at least over 80,000 publicly available data sets in just one part of NCBI database called GEO database, which stands for gene expression omnibus. Such data repositories contain datasets on thousands of genes in each sample, with tens and even hundreds of samples per dataset. Can you see my screen? I think, yeah, now it is possible. Yeah, now it is. So we discussed bioinformatics or biology acquiring properties of data science. Now let me give you a brief overview of what data science is and how it involves, how it studies different data. So at its basic level, data science involves developing methods of recording, storing, and analyzing data to effectively extract useful information. So what do I mean by that? So the goal of data science is to gain insights and knowledge from any type of data. So the data need not be of a particular type. That the data can be unstructured, the data can be structured, and data can be semi-structured. So what do I mean by unstructured data? So unstructured data is what you would have to typically deal with when you are prepared to start with analysis, or when you are prepared to start with bioinformatics analysis to make it useful. So to give you an explanation how to convert or how to go from unstructured data to a structured data, let me give you an example. So if I ask you, if I ask a poll or a question or a survey, if I conduct a survey and ask you what is your educational background, you might respond in multiple different ways. For example, I'm studying a particular subject, biochemistry or biotechnology, and you can tell I have a degree in computer science, I'm a graduate student, you can say I'm a research scientist. So somebody like a human will easily understand what each and every one of these responses mean. But if the data has to be collected from, let's say, thousands and even hundreds of students, it would be very, very hard for a human to go over this and summarize and and summarize and compare students using such responses which are unstructured. So instead, we tend to structure our surveys by replacing the actual information with short form or codes or categories which we can easily analyze. So as a result of scale, how many data points we collect, the need for, depending upon the need for the structure, the data points will, the number of data points will change. So here we are interested in identifying, or we are interested in segregating the background population or the survey population based on only 4 different data or 4 different types. So if you need to take, or if you need to have an elaborate survey, then the number of data points and or the number or the structure of the data keeps growing. So by collecting this data or by forcing structure can actually reduce the amount of information we collect. So how, as you saw in the previous category, so instead of giving an open-ended question like this, if I force you into a structure, that is going to not be applicable for many students. Some of the students might not form into one of these categories. So I don't have a category to represent them. Even including a category like others might not give me necessary information. So what happens is, oftentimes we collect data in whatever format we can, and then worry about structuring it or organizing it later. This happens as we apply different pre-processing techniques, or we perform different structuring of the data and capturing what's inside this unstructured data to categorize them into structure of our interest, and arranging data in a way where we can perform some reliable statistical analysis. So when we translate this unstructured data into a structured data, and this structured data will be this structured data is what we aim to use for analysis, interpretation, and visualization, so that it can easily be shared, so it can easily be interpreted like this. So the same would be true for any kind of data, and it is absolutely true for big data. So big data has certain characteristics that we have to keep in mind while analysis, like velocity, variability, and volume. To make sense of such data, we have to process it and prepare it for analysis. So by by what do I mean by processing it and preparing it for analysis? So, for example, to structure omics data, which is the primary form of data that we will be dealing with in this program and also in Learnomics platform as such. So we need to take the raw sequencing data, that is strings of letters that contain A, T, G, and C, and to generate a data set or to generate a matrix or generate a table of continuous or categorical data. So when we generate this table, we can subject this table for further detailed analysis, lot of statistical analysis, and etc. So to do this, we organize processing steps in sequences that we will refer to as pipelines that are specific to each type of omics data. For example, if the data is from genomics, then we have a specific sequence of algorithms or specific pipeline that takes the genomics unstructured data into structured data. And similarly, if the data is from proteomics, we follow a different set of pipeline that will take this unstructured proteomics data and convert that into structured data. So this applies for all possible omics data. Indeed, it applies for all possible data, all sorts of data in general. So the aim of using or the aim of trying to employ different programming environment for any type of biological data to any type of data is to get to a point where we have good quality structured data to begin with. So that is extremely important step. So to give you a perspective, we have so much of unstructured data and we have so little of structured data. The necessary for this conversion, the necessary for such an analysis keeps on growing. So we have not caught up yet, and because we have so much unstructured data, and people and there are ways to deposit only unstructured data, like when people sequence and when researchers sequence data, or when they acquire sequencing data, they get the biological digital sequencing data, which is unstructured data, and then they submit it or they deposit it in the repositories for everybody else to download and then analyze and then get their own biological implications or find their own biological results from. So how to do this? Or there are different ways, as I mentioned, to go from unstructured data to structured data. We will discuss few and how it is possible in for example, in the bioinformatics platform or big data bioinformatics platform that we have in Cloud Bioinfo platform. So for example, in the event of genomics data, we always start from raw FASTQ files, and then we use a pipeline, as I mentioned, called a DNA variant calling pipeline, where we select specific algorithms that will take the raw FASTQ files and then subject it into a sequence of analysis methods. And in the end, we are going to receive, or we are going to get a table of mutation or mutation information, or we'll get a list of variants that can be identified from the raw FASTQ files or raw unstructured data. So this mapping, the whole process, can either be visualized using some visualization, as I mentioned here, or the aim of this visualization and aim of this analysis is to identify those places where our sequence or the sequence for the sample that we have recorded the data varies from the reference genome that we are comparing our biological data with, or comparing the FASTQ file data with. So this variance can be listed, or it can indeed be tabulated in something like a table of somatic mutations, and such a table can be used for basic and advanced statistical analysis methods using R or Python or any programming language, even SPSS and MATLAB and anything that you are comfortable with. So basically the aim is to go from whatever digital data that we have into a table or data that resembles a table of rows and columns with numerical or categorical information that one can populate from the data that we have recorded. So in this somatic mutation table, what we have in the rows are DNA positions or genomic positions, and what we have in the columns or important column are the somatic score or the mutability index for that specific position. So higher the score, higher the mutation rate for that in that specific position, and lower the score, lower the mutation rate for that specific position. So from where we can get this table? I have a question. Aarti, we have to generate this table using a pipeline or using some sort of sequence of pipeline analysis method that I have mentioned. So if you want to get such table, maybe some publications which are dealing with this type of analysis can actually supplement this table or give this kind of table in the supplementary information. If not, then most of the raw data that we can acquire from the NCBI database or repository can be subjected to this analysis, and this data can be generated for each and every different biological sample. So let's go through. So for example, this mutational status can be often matched to presence or absence of these mutations. For example, when you are going to analyze and interpret a tumor biology, you will find large scores or higher scores for tumor sample and lower scores for normal samples, indicating the accumulation of somatic mutation in tumor samples and a mutation at normal rate for adjacent normal tissue samples. So that is the biological relevance of this data. And another example of data that we already know, that we are familiar with, transcriptomics data, which represents the mRNA expression. And this analyzing or quantifying this mRNA expression is an excellent way to analyze what is going on inside a cell by observing active transcription of protein coding genes. So this is possible, as we know that the DNA is transcribed to RNA and RNA is translated into protein. But in this process, not all of the exons are actually used for translating into protein. Some of them are skipped. And this data or this information that we can get from quantifying or analyzing the mRNA, recording the mRNAs of the cells in specific situations. So a typical analysis pipeline for the analysis of transcriptomics data will allow us to, it went too quickly. So it will allow us to clean the FASTQ files or do the pre-processing, and then it will let us to map the reads onto either genome or onto transcriptome, and then quantify these mapped reads into a table of expression quantification table, and then end the pipeline. And such a table will look like this, and you might obtain a table, for example, that has all the sample names organized in the columns and all the gene IDs or transcript IDs organized in the rows, and the intersection will actually contain the expression level that is quantified for this specific gene at this specific sample. So this is another form of structured data that we can easily use to understand the biological state of these samples differently. Usually these samples will have biological replicates, and thus our statistical power of interpreting the difference between one type of sample and the other type of sample is increased. And another type of data that we will all see in this program and also will generally is using the same sequencing technique, but it offers completely a different view of biological samples. So in this case, we are not looking at sample that contains one type of genome, but we are looking at a sample that contains millions of microorganisms and represented by their respective genome that are different from each other, that are very different from each other. To structure this data or to organize this so much of genomic information into categories, we can take genes that are functionally related and genes that are common to all the organisms and identify their divergence, or identify their differences, and identify the number of microorganisms depending upon counting this specific genes that is gene that is shared by all types of organisms. So for example, like the ribosome, which can be compared to a database of its own sequences. So such an analysis is commonly referred to as 16S ribosomal RNA, because we are actually taking that one specific gene to understand or to quantify the amount of biological, or to quantify the amount of what type of biological organism present in the sample, and it's called 16S amplicon data. And this can be subjected to a data to sequencing pipeline, And such a pipeline will actually give you or help you in obtaining proportions of taxonomic groups across samples, and this can be used to understand abundance of specific microorganisms that might be linked to the phenotypic variation. So the basic type of structured data that one can obtain from this case is also a table, and this table will contain a lot of operational taxonomical units across samples with proportional abundance in each and every cell. So these OTUs are the taxonomical variants or taxonomical groups that can be linked by group, kingdom, phylum, class, order, etc., etc. And these are the samples that will contain their respective abundance level at this number. So these are the three different types of, for example, these are the three different types of structured data that we will be dealing with over the course of this program. And this we spoke about the operational taxonomical unit that groups the input, that groups the sequences based on their similarity level into different pyramid of taxonomical order. So far we have been talking about the structured data that can be generated from at least three different sources. So now let's take this structured data and how can we represent, or how can we read, or how can we take this structured data and make a programming environment like R store and analyze this data and understand and help us understand the patterns within this data. So to do this, we have to make use of different collections of data types that one can usually find in R and also in Python generally. So I'm using this in a way where you can apply this to R and also Python because they are very similar in this case. So a vector in this case is just a collection of objects in one dimension. And a matrix in this case is a collection of objects in 2 dimensions, and at least in 2 dimensions, right? Preferably or specifically only numerical data. A vector can be a collection of different types of data. A matrix has to be a 2-dimensional, it can be 3-dimensional also, at least 2-dimensional collection of only numerical data. An array can be more than 2-dimensional in nature, and they can contain either all of numerical data or all of different types of data. But one of the most interesting and one of the most useful data type or object type that we will be using is a data frame, which is essentially a table in its absolutely natural format. So in our table, as we mentioned, if we have to prepare a table that gives structure to some sort of data that we have collected, in that table, we will be inclined to include, for example, a character type that can represent your name. It can include, for example, a numerical type that can represent your height, weight, etc. It can include, for example, a factor type or a categorical type, like your blood group, which takes only a specific set of variables, not all possible variables. It can take something like a date type, like your date of birth. It can take something like a logical type, whether you are male or female, that can be converted into binary logical database. So this, what you see, is a collection of different types of different data types or different classes of data that can be organized into one table. And such an organization, preserved as such, will represent a data frame in R or in Python's language. So we will be using most of this data frame, and we will load all of these structured data that we have been discussing in the previous slides into a data frame, and then we will start our analysis from that. So in addition to this, you will also find lists, which is just collection of different types of data into an organized group, so that you can query them, or you can access them whenever you need to access them. So what can we do with such a structured data that is loaded as an objects in environment like Python or R? So for example, you can run some a transformation analysis, you can run analysis on the distribution, so that we can understand what's the pattern of the data, and we can understand if there is any specific correlation, or if there is any specific relation exists between two features or two genes in the data. So if you want to identify whether gene 2 and gene 3 are correlated, we can use this scatter plot for gene 2 and gene 3 in specific, and then identify their relationship to each other. So what is FPKM? FPKM is a normalization applied to transcriptomics data, which refers to fragments per kilobase million. So when you count the mRNA molecules that are mapped onto different genes, then these absolute count can have influences or can have biases due to the length of the gene and the coverage, depth of the coverage. Yes, it's one of the normalization means, and the depth of the coverage that gene will actually encounter. So FPKM will eradicate or will actually account for that difference in the coverage due to the length of the gene and also due to the depth of the coverage that this gene can encounter. So we can understand the data further and use different visual representation to spot technical noises that is important to eliminate, and also to spot to spot specific signals that we have to concentrate on to understand the patterns in the data. So all these analysis we will perform in the upcoming sessions. So we can also extend this to more biologically meaningful data analysis and exploration, like a heat map, which will give you a generic, a general expression pattern for different types of samples that you have in your data set. So we can further extend this to much more advanced statistical analysis like data mining, classification, supervised machine learning and unsupervised machine learning, to understand or to explore the relationship between the features or genes to understand the genetic pattern that is present in our data, so that we can relate or we can correlate this pattern with the biological nature or the phenotypical characteristic of the data. So this is what we are going to deal with in this program so far. So if you are through with this, let me quickly go to the lessons and give you a demo of how we are going to go about these analysis methods and etc. So if you have any query, please put that up in the chat. Let me stop sharing my PowerPoint presentation and then get to my browser. So do you have any queries so far? Please mention that in the chat room. So let's get, where is my, this has to be it. Please confirm you can see my screen. And what is the screen that you see? Can you see the loading data in our screen?
Yes.
Okay, thank you very much. So so this is one of the associated lessons that you get. That you get with your in your program page. This is where I took it from. So these are the associated lessons that we are going to discuss so far here. So if you go to Introduction to R, you will get an introduction to what R is about and then how to get R. So how many of you have installed R in your computer? Do you have access to R Studio or R in your computer? Please represent that. Please let me know in the chat. Yes, that's perfect. Then if you have R in your computer, please open up this lesson, both R and R Studio. Perfect. Then R Studio is more related. So open up this specific lesson in your browser window. You can follow along as we explore this lesson. So the first, excellent. So the first section is actually going to introduce you to R as how to download and how to install in your computer, and how it is generally useful for bioinformatics. But I'll let you go through this and then skip next to getting started with R. So here you see we take this specific example code, which is a code to read a table, or which is a code to read a structured table that we have been discussing so far, and then read this table into an R environment. So let's copy this, and then I'm going to go. Let me share everything. I think I have to share everything so that you can see my R Studio also. Yeah, I think you can share. You can see my screen. Confirm it.
Yes.
Yeah, thank you very much. So your R Studio window will look like this. So this or the other way, it will look like this. I have it organized it slightly differently. So your console window will look here. Let's do that sort of organization, and console on the left, console on the right. I should pan layout, go there. Where is this gone? Okay, here, okay. Okay, this is how it is going to look like for you. So what do you see here is, let's apply, yes, what do you see here is you will find this is the R console where we are going to interact with R in this window, and R is going to give us what it is going to process from this window. And this is where you will see all the variables or objects that we are going to load into R is going to be displayed here. And this is where you are going to see your plots. You can see your plots here that you are going to generate, and you can see what is the current working directory and what are the files available in the current working directory. Yeah. So the file or the line that I just copied can be pasted onto what we see here as the source window or source panel. This is the source panel, and this is the console panel. So in the source panel, it's like writing an R script where you can write the R script and then execute this R script line by line. So it reads, it tells we are going to use a function read.table, and also it tells you I have to use a specific path to find where to find this specific file, and also I have to use mention several other parameters that is going to describe the data that I have. Okay, let's go back to the lesson, and let's see what the lesson tells us. The lesson is going to tell us, give us a link to where to find this specific file name. Well, let's see, where is the file, associated file? Okay, it's not here. Let's get the file. So in order to get the file, you can actually come down and get a link from here. So this link, if you open in a tab or a new tab, is going to open up like this. So this is a structured table, which is a gene expression table, and then what we can do is we can copy this as a link address, and I'm going to paste this link in the chat window. If you have opened this, you can follow along. You can copy this, and then you can go to your R Studio, and then right between where to find this path, right? You have to delete this and then paste it. Exactly in this position, paste it. So this R can also read files that are made to be available or made to be accessed through internet. So this is a file that one can access from internet. So that's what we are going to read. So let's break this down, right? Let's break this read table or loading this this table into R a lot. Like, let's just read the table without anything, right? Read.table, and then type this. So what am I typing? I'm just using the function read.table and then specifying the specifying the table where to find that specific table. So if I execute it, wow, my code runs. It actually got executed. But what do I see? I see an on-screen output, which is, on-screen output of how R had actually read this table. I do not see any variable or any object that is stored in my object or variable window. So to access this information or to access this table, we have to store whatever we executed right now into an object, so that we can keep retrieving that object and use that object for further statistical analysis. So that is what we are doing it here. So R uses this specific operator, which looks like which is a less than symbol followed by a hyphen, to redirect whatever you see as an output of this specific function into this specific or this object. This object will be a data frame, and that is why it is abbreviated into DF. So let's do that and increase the complexity one by one. So let's do that. And then this is going to be read.table at its absolute minimum. And then do it. So in order to execute this, you can select all of this and then run. Or you can keep the cursor at anywhere on this line and then run. So I am keeping the cursor on this specific part in this specific line, and then I'm running it. So if you see carefully, there are two changes that happen. So this line executed successfully, and R is not giving me any error. And in addition to that, I'm having one data or one object that appear in my environment page. And this tells that this object is 16 observation and 53 variables long. So what do I mean by that? If I click on this, I will be able to visualize this just like Excel format. It is not advised, it is not advisable to do that, but for small data set, it is okay. So what do they mean? 16 observation would mean 16 rows, and 53 variables would mean 53 columns. That is what it does, it means. But if you see how our data is actually visualized, there is a difficulty, or there is that's a disadvantage. Our data actually has what we call as header. Header is what we in our data that specifies what each and every column would mean. So this column means ID column, this column means normal like column, and this column would be normal like column, and this column would mean vessel, and etc., etc. But by mentioning nothing about what our data contains and how our data is organized, we have actually encountered a problem where the header is actually read in as one of the lines, and that is going to obscure, or that is going to not be very helpful for us to perform any statistical analysis on this specific data type. So let's correct that by mentioning one additional option or one additional parameter which says header equals true. So that would generally mean we have a header line in our database, in our input table. So I run this again. So now you immediately see, since I ran it and since I have put it or edited the same line and I redirected the output to the same object, the object became 16 observation instead of 15 observation. So let's view this now. Now it looks like a little bit better, and okay, let's compare both. Let's do that so that you can understand. So I'm going to rename this as DF1 and keep the old one as DF, so that we can do a side-by-side comparison. And then I'm running the old one first, DF, and then I'm running the new one, the DF1 next. And then let's visualize both. So this is the visualization of DF, and this is a visualization of DF1. So what do you see? What is the change that you see? Can anyone spot the change in the numbers? Did you get this far? Did you get this far? Did you try to run this and did you get this far?
Yes.
So what is the change in number that you see in the numbers?
Yes, the observation has changed from 16 to 15.
Yes, the observation has changed from 16 to 15, but if you see here, the zeros are just zeros and they are left aligned.
Yes.
Right. And here you can see we have right aligned and the zeros have their significant values. So if you have worked with Excel, you would know that anything that is left aligned is a character or it's a text format. So in spite of these values being numbers, since the first value is a text, all of these values are considered as text only. So in this specific data set where we mentioned the header, where we did not properly describe our input table. So in this function where we describe the input table properly, all of the numbers are read in as numbers. Okay, we can get some more information regarding this. I will demonstrate this to you in the next session, where we can perform some summary operations, some statistical operations on these numbers, for which such a detailed or such a level of description of the data is very helpful. So we cannot perform any statistical analysis on this. We cannot calculate mean, we cannot calculate sum, we cannot do anything in this way. But it is possible to do that, which is our sole aim, which is our main aim. So what does the zeros in DF1 mean? So the zeros in DF1 mean there is no expression for this specific gene at this specific sample. So that is what biologically this one means. So gene 2 is expressed at 2.04 times 04 FPKM value for this specific sample. And similarly, gene 2 is expressed or is not expressed at all in the luminal sample. So this is what zero biologically means. So describing the data correctly becomes extremely important. So in addition to this, we have some more parameters that will become extremely important for us to move forward. For example, what is the next parameter that says? The next parameter is the one that says how each of these columns are separated in our input table. So if you open the input table in any notes or in any text editor, you can see that the columns are separated by a tab delimited value. So that is what this one says. So read.table by default, right? By default, it assumes that your data is tab delimited, and that is the reason our data is read in correctly. If our data is not tab delimited and if it is a comma delimited, then there would be a big mess in the data, and we will not be observing these many different columns in the data. That is what it is. So what else do we have here? So we have here row names equals 1. So let's take this, use both of these, and go to DF2 and do this and see what change does this actually create in our data set. So if you run DF2, so what changed? What did change here? The number of columns reduced from 53 to 52. So let's open up DF2, which is here. Let's put it here. So what happens here in DF1? Observe what is mentioned on the left. This is the row name or row number or the quantity or the identity with which this row can be called, or this row can be identified with, or this row can be selected for further statistical or further different analysis. Now, by including the flag or including the parameter row.names equals 1, right, equals 1, then what have we done? We have changed the first column into a row name. So the first column is now known as gene 1 column, gene 1 row. The first column will actually replace the row names, and the first row, instead of being known as 1, 2, 3, 4, 5, 6 in these two data frames, it will be known as gene 1, gene 2, gene 3, gene 4, gene 5, gene 6 in these ones. So that is what it is. So that is why it is very important. And the next one more phase. This will be important in the case if you have, for example, competing row names or competing column names. If you have that, in this data we do not have that. If you have that, R is going to throw you an error, say that you have duplicate row names or you have duplicate column names, I cannot load this properly, you have error in your data. So having this specific parameter will actually turn down that check and then let R load the data in whatever format it is, so that it is read in properly. So that is how you can load a data, import a data into R code. Since we are going a little bit overboard, this we covered. So this is not restricted to a .txt file, and this is not restricted to columns that are separated by comma. So this is the same logic, and I want you to go through this associated lesson where we have multiple different options, where when you use option like semicolon, then you are using a file with a semicolon separated format. But more, for example, related format or more relatable used format is CSV format, comma separated file format, which we can use for these analysis. So let's see if we have a CSV format in this one. So I have demonstrated you how to take this code, put it into RStudio and try to run it. But if you do not have RStudio, or if you do not want to change to a different environment, you can do it here itself. This is an R console. We give it to you for you to practice whatever you have learned from your in this section. So in this section, as I mentioned, you are going to go through these functions or these different codes that will help you import on viewer text file into R. So that is what it does. And this tells you how each and every different parameters actually correspond to different options, as I had actually mentioned, and how they describe our data in its entirety, so that we can load the data in the form that we want to load in, and to perform necessary analysis like this. We will come to visualization in next lesson or the next session, because right now we are running out of time, and I quickly want to tell you how to use or how to use this console for your analysis. So read through this specific line. It says here we deleted some parts of the syntax of the code, and you need to complete those parts so that this R code can run successfully. So if I run just like this, it's going to throw you an error. And yeah, it says it cannot open something, and it's going to throw in error. So what is missing? The link for the file is missing, which is what we can find here. You can copy link and then go to this specific path and then update that with the link, and then try to run it. If you try to run it, it will give you different error. Okay, why does it give me different error? And it also tells success in running the code, but the answer is incomplete. But if you had if you have gone through the data, if you have gone through the lesson, it will tell you there is an important function called dim, or dimension, short form for dimension, dim function, which will tell you how wide and how long your data set is. So let's run this. Now it says success running code. The answer is correct because it just keeps on updating. So we will clear the console like this and then run it again. So now it says this data frame that we loaded has 15 columns, 15 rows and 52 columns, which is what we saw in RStudio here. So the same dim function can also be used here to identify or to see, to verify what is the dimension of this specific data frame. So it is like this here, 15 and 52. It's always rows first and columns second. So the dimension of this data frame is 15 rows and 52 columns. So that is, so you can practice that in here. If you go to next section. Next section deals with how to visualize box plot. So all of the incomplete code that you will find in this import data, in this test your skills or in executing R code can be found in this learn the code section. So if you go to learn the code or go through the learn the code section carefully, then you will be able to relate to this. You'll be able to relate to the necessary codes or necessary completion of the code that is absolutely required for you to run this code. So this is so far. This is what I will cover in this class, and then for the next few classes, we will go through. We will discuss after loading the data how to perform some really starting sample statistical analysis and summary analysis, and how to visualize the data in the form of box plot or a scatter plot or a heat map, etc., so that we can understand the general pattern that one can identify from the data. So if you have any questions until now, please let me know. If not, I see in the chat that some of you said it is not for, you did not get to the same stage as what I got. So maybe if you go again and go through the associated resources, and it will be helpful for you to recreate, or be helpful for you to execute the code in RStudio, or you can copy it and execute the code in the try it for yourself section just below the learn the code section. So thank you very much for now, and if you have any other queries, and let me know, and I hope to see you, all of you, in the future sessions, so where we are going to not only just load this data, we are going to do some lot of summary about this data. We are going to print out summary statistics, and we are going to see the distribution of the data in terms of heat map, density plot, histogram, box plots, everything, PCA and everything, so that we can learn more about the data, which is what our aim is. If we have no more queries, then I can pass this on to Sonalika. I think there are some questions about where to find recording of the session and where to find slides for these sessions. I think Sonalika can let you know.
Yes, so the registered participants will be getting the recording of the sessions. RT, I don't think that you have enrolled yet. So I'll just quickly take you through the program page so that you can proceed with your program enrollment to get the access to the recording and also the other sessions. So today we had the first session, and I thought that we can write our registrations as well, so that you know how these sessions would be conducted. It was just an introductory session just to get you started, and we'll dig more deep into the details of the topics in other sessions. So now I'm just going to give you a quick demo sort of thing to tell you how you can register yourself for the program. So here is the link to the program in the chat. And those who are interested to enroll for this particular program can pick up any of these 3 subscription levels: beginner, intermediate, or advanced, and get started with the program. So in the beginner level, you'll be a part of such sessions, and in the intermediate level, you'll get the access to scheduling one-on-one sessions in addition to such sessions. And in the advanced level, you'll get some extra time, around a month or so, to work on your research proposal and get started with your research project, right? So these are the 3 differences in these 3 subscription levels. And if you are interested to pay beginner, intermediate, or advanced, that's your wish, and you can get started. Yes, the class was recorded, and we'll be sharing the link of the recording with the registered participants, Lisa and Louis. All right, so now I think we are all set to get started with another session on Python. So I think the specific participants of Python will be joining in some while. And so we discussed about, it was a a wonderful session, and I hope now you have a sense of various types of omics data types and why do we need to analyze it, what type of data do we get in these different sorts of fields. And today we just learned about how to load data and just work with data in like got to know about some few concepts that would help you. And in the next sessions, we are going to discuss about statistical analysis and visualizations as well. Now let's get started with Python. And again, the same thing. Here is the link to the program page for getting started with bioinformatics in Python. On the page, you can see that we have all the sessions scheduled, and you'll be able to look at the session recording again on the program page itself. And the access to the program page is given only to the registered participants. I'm repeating it just to make sure so that you know who all will have the access to it. And also then you have the resources that you need to go through as a part of each and every session. So today we discussed about just getting started with our Dr. Raghav took us through the 11th lesson in the bytes and the molecules coursework to get you started. You need to complete that, and also introduction to bioinformatics. Similarly, for our today, we'll be completing loading data and data visualization lessons as a part of the very first session. After the session is over, I'll put up the recording, and also you can revisit the recording and click on the complete session icon once you have watched the recording, completed the sessions. So that was about getting started with Python. Now I would like to pass it on to Dr. Raghav to help us get started with Python.
So before going forward, can I ask from the participants, do we have any new participants for Python? Or if you are the same ones, if you already, all of you who are present here have been present for the R session, then we can quickly go straight to Python session instead of all the same introduction that I have planned for the Python.
So, I think most of them are the same, and also more participants are going to join us, and those who will be joining us, they can go through for the understanding of big data and various types of data types, maybe the recording itself from the very beginning, and you can, I think, get started with Python directly. What do you think about it?
I mean, if that is the case, then we can continue with some more of R and then include Python also, so that it completes the session. We can show them some visualization. We can show them how to retrieve the data and continue if that is the case. And if we do not have any new participants, so we can hear from the participants themselves. So how many of you participants are here for the Python session only? So how many of you have joined only in last 10 minutes? Can you please give me a yes or one in the chat box? I think most of them have already been here from the R session. If that is the case, then that is excellent. And then we will continue for the R session for another 10 minutes or 15 minutes, and then go to Python and how similar data loading can be done for Python also. Okay. Will that work for you? I think that will be good. Yeah, because if most of the participants are same, then they can access the introduction to omics data similarly. Yeah. Okay, let's continue. In that case, then that's excellent. So let me share my screen again. So I showed you until now how to load R file. Okay, I have a chat. How to load a data table into R as a data frame, and then how to use different parameters so that we describe the data frame, describe our data table to its fullest, and then extract a lot of information from the data frame. So what do I mean by that? Let's go back to the R Studio, and if you are following along, please keep your R Studio also handy. So now we saw that using different setting parameter settings, we can describe the data in a way how we want it. And once this is done properly, once this is done appropriately, and then we take that specific appropriate data frame, and then we can subject to multiple different analysis. So I showed you to demonstrate you the difference between these three data frames. I just clicked on this data frame and then viewed their view on here in the source panel, or in the in the view panel or source panel. But this is not going to be possible, or this is not going to be efficient. This is not an efficient way of displaying or skinning or actually getting an introduction to your data frame, because most of the time when we are going to deal with data frames or tables at the size of thousands and thousands of rows and hundreds of columns, this line or this mode of viewing and exploring the data frame or object will actually slow your computer down, will make your R run inefficiently. So in order to do that, we can start with simply typing the name of the data frame, for example, df2, and then executing it. If you do that, so that is going to execute, that is going to make all of the data frame visible, or it's going to type, or it's going to make all the data frame, print all the data frame on the on-screen window. So this is not going to be useful for you, and for this data frame, which has only 16 observation and only 50 to 16 rows, 52 columns, it was possible for me to scroll up and then check where the beginning and how it looks like. But for most of the cases, this is not going to be very, very informative for you, because you're going to keep running for thousands and thousands of rows, and this is not a way, this is not a good way or efficient way to explore your data table. So what is a good way to explore your data table? So let's use a function called head. Head will actually take in any variable or any object into its as an input. It's a function that is going to print only first few lines of your, or first few rows of your object. The object in this case is the data frame that we have loaded with our table. So if I execute head, see what happens. It is going to give you, or it's going to show you or display only first 6 rows of your data frame. And even then I have to scroll something like multiple times, because I have all the columns. Head is generally going to display all the columns and only first 6 or 5 rows, depending upon the setting that you have set. Most of the time by default it's 6. So how to get around this displaying of all the columns? In order to do that, we have to make use of how we can refer, or how we can refer to different columns and different rows of a data frame in R. So if you can follow along, and if you can make this execute this in your R Studio, please go ahead. And if you, I'm following the chat. If you have any difficulties, please let me know. Now we know that data frame is a collection of rows and columns and of different tables. So how to access, for example, first row, only the first row, or how to access only the first column? So that can be done by indexing, what we call in R languages, indexing the data frame. So, or by exactly mentioning or specifying which row or which column that we are going to access, or that we want to take from this specific data frame. So that can be done by using the square bracket. So R has this feature, or not R, R Studio has this feature. If you open a square bracket, it will close all by its own, right? Inside the square bracket, as I always mention, it's always beginning. I mean, the first index is always row, followed by the second index is always column. So what is the first index here? It's a row. So if I want to display the first row of my data frame, I type 1, and then if I run this, what will happen? Can anybody guess? Can anybody guess? Will this code be successful? Yes or no? Simple. Will this code be successful?
Yes.
You can type. Let's try that. Let's try that. Let's run this. Yes, it is successful because, sorry, I have to type df1, df2, f2 of 1. Yes, it is successful because of the way how R is going to treat your data. So by default, it is going to assume that you are mentioning a column of this data frame. If I want to have the row of this data frame, I have to mention row, comma, right? It's always row, comma, column. So if I have to take the row of the data frame, I have to mention, I want to take the first row of the data frame, and I want to print all the columns of the data frame, and hence I have kept it as or left it as empty, or left it as unmentioned or unspecified in the column place. So if I run this specific, yes, in this specific code, it is going to give me the first row in multiple display rows, first row and all of their columns. If we go to the display, you can see that the first row, which is named by gene 1, is getting displayed. So it's 0, 0.454, 0, 13. That is what is getting displayed. This is how we can call for specific row. For example, if you don't want to use this confusing way of naming of indexing of column, then we can usually use df2, similar to what I have mentioned here. We can leave a blank for the row, and we can mention the first column. Sorry. And then if you run this specific row, it's going to give me all of this in a list format, or in a format where it is giving, you observe the difference between the format. And this format is like a example data frame format, right? This format is like a list format, right? Just printing all the values one after the other. Both these formats will have their place, but in general, we will prefer indexing like this, or selecting specific rows or specific columns like this. Like, okay, if I have to mention more than one row, if I have to mention more than one row or more than one column, how do I do? So since in our data set, the interesting bits are the samples. Let's take the interesting samples, like rows are also interesting, genes are also interesting, but we are generally more inclined to understanding the sample distribution. So we will take or we will actually try to fetch or try to display first two columns of this data frame df2. So how do I do that? So you can do that by using the sequence operator, which takes a colon in between. So 1 to 2 will mean, let's see what it does. 1 to 2 will generally mean just mention 1 and 2. So if I type 1 to 10, it's going to take, look, can you see that? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is a sequence operator. So from 1 to 2. It doesn't have to start with 1. I can start with 5 to 7. So it's just 5, 6, 7. So, but it cannot, it cannot go in reverse, I think. Yeah, or it can also go in reverse. So I was wrong. So yes, so you can use 1 to 2 in this case and then run it. We are going to get both first sample and the second sample as our subset or as our selection. Such selection methods will become extremely important, because when we want to select only specific samples for further analysis, if I want to check only specific samples for further analysis, this is how I will do. And if I want to check only, let's say, first 10 genes of all the samples, and this is how I will do 1 to 10, comma, and everything. And if I run this, this is how I will do. See that I'm getting first 10 rows or first 10 genes of all the columns or of all the samples. So what if I want to take first 10 rows and only first 2 columns? So I can combine these two, df2, 1 to 10, comma, 1 to 2. So this is what, if I run this, so I'm getting first 10 rows and first 2 columns of these two. So this is how you can index, or you can mention with the numbers what are the different columns and what are the different rows that you have to, or that you want to mention, or that you want to subset from your sample, from your data frame. So this is about exploring the data frame in the environment of R. So we already had an introduction to head, and we already had an introduction to dim, dimension, output. So dim is just nothing but it's a short form for dimension, and that is going to give you the dimension of your data frame. So it says this data frame is 15 rows long and 52 columns wide. So that is what it's going to do. So how many of you, since we are also going to deal with Python, I want to tell one very important difference between R and Python. So if you see here, in order to query, or in order to fetch the first row or first column of the data frame, I used the number 1. So I use the number 1. That is R's convention. All the index or all the arrays, matrix, vectors, everything starts from 1 in R. But this is not true for most of the programming languages. As for Python, it will start from 0. Okay. One easy way to remember this is, how many of you are from US? In the US, we do not refer to the floor at the level of the ground as a separate 0 or ground floor. We start with the floor 1. Floor 1 would mean floor that is at the ground level for the United States. That is R's convention. In Python, which follows something like British, or which follows something like British occupied other colonies, where we have a separate floor or 0th floor for the floor that is at the ground level. So it's 0. It always starts with 0. So for the same indexing that we are going to do for the Pythons, in the Python's demonstration, we will start with 0. So the first row will be indexed by 0 number in Python. So this is one important difference that you will have to remember. And another one, this operator is specific to R, which takes whatever is on this side of the operator and assigns that to this side of the operator. So this side of the operator can only be an object or name of the object. So this side of the operator has to give, or has to result in some sort of on-screen output. So it will take this output and then redirects it to this specific operator. So this is also very unique to R. This can also go in the other direction also, but we don't want you to confuse, so we just stick to this. This is equivalent to this one, equals df2 equals R. This one will also give you the same result, but since this equal to operator is used for other purposes in R, so R has a specifically different operator for this redirection and assigning whatever is on the output to a different object. So this is what I wanted to show you. So in the associated lessons, you will also find some this for box plot. So you will see that. So if I type box plot, let's try that, of df2 and see what happens if I run this code, and what happens to my RStudio if I run this code. So I am going to get a box plot in this plot viewer. Can you all see my plot viewer where there's a small box plot that I have generated? So let's zoom in. If you zoom in, it's going, yeah, it's going to show you in big. So we will learn more about how to change something like this. No, can you not see this screen, Michael? Michael is having difficulties in this. Can you confirm that you can see this screen where I have taken this box plot and made it a little bit big here in the plot zone? Okay, thank you very much. So yeah, so we will learn more about how to make this plot look better and more informative in the terms of populating it with a lot of different sample names and coloring it accordingly, and then transforming the scale to log, etc., etc. Excellent, very good, Emmanuel. So in this one, so for now we will leave it this way. So do you also, all of you also remember the difference between these three data frames that I was talking to you about? And specifically at the beginning of the session, I was talking to you about this data frame, which had or which did not represent our table properly and considers all these numbers to be characters. So how can we check that? This is one way to check that. If we use df instead of df2, I urge all of you to try that. df, and let's run this. What does it say? Yeah. So what does R say? Congratulations to all of us. We encountered our first error in our programming language. So nobody can escape encountering error. But what does it say? It says non-numeric argument to binary operator. So it cannot deal with non-numeric argument. In spite of most of the data frame or most of the data set having numbers, it starts with, or the first data or first entry of this column or of every column is a character. All of these numbers are treated as characters, and hence box plot cannot deal with character data. Box plot has to deal with, box plot is an analysis that takes a numerical input. So that's what R is telling you about. So now that we have extracted data from df2, where does the original data go? No, it is not lost. We have just extracted it here, no, right? So we have not, just like how we did for the first read.table function, we have just made it display on the table, on screen output. If you want to say, for example, this subsetted data set into something new or something, some other object, so we have to specifically do that. So let's do that. So that's a good point that Greg brought that up. So let's do that. Since we are successful in identifying our interested subsection of the data frame, so that data frame, I have a habit of having the parent data frame followed by the underscore and whatever modifications I do. So I started with df2 and then I left with something that is small, so s. So that takes whatever output. Yes, exactly. That takes whatever output this is. So df2 1 to 10, comma 1 to 2. So if you execute this, so that is executed, and you see here we have 10 observation and 2 variables, or 10 rows and 2 columns. That is what we observe here in this specific one. And yeah. So both the data survives. The data to df2 from which we have subsetted this also exists, and df2s for where we have taken this subsetted data and then assigned it to ds2 also exists. So if you take this, so this is what it looks like, and df2 goes here. So let's put it here. So df2 is a whole data set that also survives. df2s is a data set that also survives. So until or unless explicitly I mention to delete df2, or I reassign something into the same name, whatever once loaded will not disappear. So it will only disappear if your R session gets rebooted, or if your R session quits or something like that. So whatever once loaded will stay here as long as you quit the session or end the session. So I hope that is more informative. And if yes, then we can move on to Python, how Python's way of dealing with this data table. Excellent. Thank you very much. So let me stop my share for RStudio and get your Python lessons in order. What is this? Okay. Okay, let me share my screen. Okay, I have to share everything. That's okay. So, how many of you have are using Windows machine? Some of yes, some of you might use Linux, and some of you might use Macintosh or Mac machines. So, yeah, for people who are using Mac machines, Python is going to come pre-installed in their computer. Only the Python is going to come pre-installed and not the interactive development environment, as we would like to call the R Studio that I have described you, that I have been using to show you how R can be run in a very user-friendly environment is called IDE, Interactive Development Environment. So for many of these coding languages, we have to use some sort of IDE so that our job becomes easier, because most of the time we are using R and Python for visualization purposes. So in the native R and Python consoles cannot produce a visualization output on screen. So that is why we have to resort to this IDE. There are several IDEs available for R and Python, and for R, the most used IDE is R Studio. Yes, sure, Mary. Yeah, you can do that. Yes. So R Studio is what you will, most of us will use to run R in a user-friendly environment. And for Python, we have several of at least 3 different ways in which Python can be interactively used, or interactive development environment of Python can be used. So one way is by using Visual Studio Python, or Visual Studio code for Python, where you can try to download VS Code and then launch it. Then you'll have a very user-friendly mode of running Python. Another way is to use Anaconda or Coda libraries, where it is going to install a set of interactive development environment for not only for Python, for R, and for other different coding environments in your computer. So that is going to be big, and this Conda is going to be helpful for many people. So because it is going to install its own version of Python and R, which you can manage it easily, so that you can check it out. But one of the easiest way to run Python is using a Jupyter notebook that is online, that is available online in Google Colab format. So that is going to look like this. If you have Google account, you can use, or you can leverage Google's resources like Google Colab or Collaboratory, which is a Python notebook where you can run these Python codes on the Google Colab in interactive method. So that you can also do, and or you can follow along the Python's sections or Python's codes here in the learn the code section, where I took it from your associated or learning resources in the in your program page. So this is where data visualization and loading lessons for Python is where I took this specific lesson from. Let's see. I will share this lesson with you in the chat room, so you can again open it for you. Okay, somebody asked which one would be better if we want to use offline. Which one would be better if you want to use offline? Anaconda or Conda libraries are going to be bloated up. It's going to be a little bit big. So if you have some space, you can use specific Condas, or if you want to use only Python and some development environment for Python, then and you are mostly in Windows machines. So install Python as a standalone and Visual Studio as an IDE for Python. That is going to be sufficient for you. So that's going to helpful. That's going to do all the job that you need. And if you have space, and if you have much more freedom, or much more not freedom, much more organized way of dealing with libraries and packages and all of these installed libraries, then Coda is the one that we want to check. let's see. I mean, I'm also not sure if I think I've seen this Conda. Let's check. And yeah. Yes, it is available. It should be available for Windows. Yes. Yeah. So this is Docs Anaconda. So Anaconda install for Windows is what you want to check. And this is how you can install it for Windows and get the necessary district. So it's going to be a little bit bloated, but it is going to be very, very helpful. This is going to, Anaconda is going to install you a separate IDE for Python. And it is also going to install you something like Jupyter Notebook for Python, where you can run Python from your computer on Windows or sorry, not Windows, on the web browser environment, just like exactly like Google Colab, but you can use it for offline and you can use it for your own from your own computer resources. So that is one of the best way to run Python. It involves letting, yeah, exactly, yeah. VS Code is also better. It's very simple, but Conda installation and setting up requires a little bit of finesse to make it work. But if you if you know, or if you can do a little bit of modifying here and there and understanding how things are getting installed in your computer, then Conda is going to be worth a dimension. Okay, let's go to this one. So this is going to deal with only Visual Code and this Python collaboratory notebook. So how to access this Python collab? You can actually, okay, loading. Let this, we will let this, okay. Reconnect. So if you go to Google and type in Google and then it will give you this welcome to Google Collaboratory. So it is just, yeah, you can click and then you can go to collab.research.google.com and then run the collab notebook. Let me get to the link and then share it with you. I think there should be a very easy link. Yes, okay. It's actually telling me that I have several of the collab notebooks available. So new note. And let's get the link from here. Yes, I got it there. Yes. I think this is what we want. Okay. Copy, and you can use this for Python's this, and then so this is, you can generally go through what this is, and oh, they also give you some example codes also. That's also very good. So let's go to the lesson and say what and see what the lesson deals with. So the lesson should actually tell you what are the different ways you can access or you can run Python in your computer. And then again, we start with learning the code. So for most of the purposes, it's going to be same. Like we have numerical matrices, just like how we have a numerical matrix for R, and we have a data frame, just like how we had data frame for R, and we have different packages or modules that we have to import in for example, in Python's environment. In R also, we might have to load some specific libraries if we have to perform some specific functions that is developed only for that library. So more on this different libraries and different functions and usage of different codes related to specific to those libraries we will deal with in the upcoming sessions for R. But you cannot work with Python without loading or without importing at least necessary packages. So all of the Python code starts with at least a couple of packages, like we want to import the pandas package, which is defining how a data frame is going to be defined or used. And we want to import something like the NumPy package, which is going to define how matrices and different matrix operations are going to be used. So again, we know what is matrix, and the matrix is a collection of numerical objects that is in columns and rows, and pandas is a collection of different data classes or objects of different data types in a tabular format. So how to do that? We have to import the package as some package so that we can call np to mention specific what do they call as attributes or specific functions that are defined in NumPy library. So let's open up this Google Colab. It's all very this for me. Let's open up a new notebook so that it's new. Yeah, this is how you will see. So this, I think it's better if you can open this specific notebook in Python, let it load. Yes, so this is what you will see. So let's take this and put it in the chat box. Where does the chat box go? Oh, it's there. Okay, so this is what you will see. Okay, let's take this import NumPy as np, and then import pandas as pd. So you have to run this. How do you run this? First, we have to connect it. So everywhere you see this connect, click to connect. So you connect it and wait for this to allocate the memory resources and allocate the resources, and then perform this connecting this thing, perform the connection. Yes, and then we have our own Colab ready to be explored and work with. So you run this. If you run this, what are you going to get? You're not going to get any output, but you're going to get a tick mark that says this code is successful. Then let's open up another text or code. So text is going to give you a window where you can type the text, right? Like this. Okay, this code got created. So this is the text that we had. Okay, this is the situation that we had. Let's open up a text window. This is a text. Okay, let's click outside. This is what it will look like. So we can click another code window after this. So this is what it is. So you can document your progress or document your process of running through this codes as you wish, and so that you can share it with your peers. So this will be extremely helpful in sharing codes. So if you go here in the Python, then again we will load this object into Python. So again, it's simple. Just like read.table function in R, we have a function here that says read_table, and we will need a file name for this, or we will need a path for this one also. So let's copy all of this and see what do we have here. So if you are walking along or working along with me, so do that. We have the link for this in the same one that I shared with you, or you can take it from down here, down here in try it yourself section. That can also you can take, or let me get this from instead of using some specific or some generic output, let's take more relevant biological data, which I showed you to describe, which I showed you to describe in which I described you in our coding session. Let's take this one and go there. Are you following along? Yes, here. Okay. So if you execute this, it's going to load, execute, and then it says execution successful. So similarly, just like R, we already saw, we have to load this, or we have to run this input, or we have to run this function and then assign it to a object, to an object. And the object here is again a data frame. So how do we know that it's a data frame? It's probably, it's mainly by default, it's a data frame in R, but in Python, since we are calling the read underscore table from pandas library, it's a data frame. Pandas defines a library, so it's a data frame. So you can type df to actually show first few lines or show the show the contents of this data frame. It's going to show it like this, and it's going to be similar. It's 15 rows and 53 columns, exactly like that. But we see some changes. What is the change that you see that is not that you do not see in the R environment? Do you remember when we run this specific code in R's environment? R had a default situation where the first line is not treated as a header. It is treated as part of the table itself. Do you all remember that? Yes. But in Python, that is changed. So Python is a little bit more user friendly, as it actually assumes that you have a header information in the first row, and it takes that header information for that, and it also assumes that your table is tab delimited, and etc. etc. So Python is yeah, in that way Python is more intuitive and user friendly in that case. But you can also read in using the same separator function. You can also read read the other delimited files. If your file is comma separated or comma delimited, we can use comma delimited also, but we have to use the separator, which is exactly the same in R, sep equals tab. Okay, but what if our data set does not have a header? So again, just like what we had in R, our data set here has a header, so it does not matter. So what if our data set does not have a header? So in that case, you have to mention again the same flag, but instead of true or false, you have to mention it as 0. So if you do that and run it, then it's going to run and it is going to complete. And if you run the df, and now, okay, why is this not working? Suddenly it started to tell me that your header equals 0. Why is yeah, for some reasons it's not working. Okay. Let's try false. False is not the type. Okay, loading data. I actually had this somewhere. Yeah, okay, let's leave it that way. I don't know why it is not working if header is mentioned as 0. Let's say 1. Yeah, okay. Okay, it says header equals 0. Okay, that's not the case. Header equals, okay, header equals 0 is not the case. I don't know what is the option for no header. There should be an option for no header, but yeah, we will leave it that way. I will find out and tell you in the next class. I also have to find out. Okay, so unlike R, Python is more user friendly and intuitive, where you see that if you type the data frame in its entirety, R is going to keep on printing everything until it stops. But Python doesn't do that. It runs for first few rows, and then it takes an extension operator or tells you that there are more information here, and then gives you last few columns also. So you get a decent understanding of the size of the data frame. So this is how it is. Again, in Python's case, let's go to the lesson. You can modify this separator to load a comma separated format, and also you can load also Excel file. So read underscore Excel can read Excel file, but usually this is not recommended because we will do multiple different operations and multiple different changes in the Excel file, and reading it from Excel file is not recommended. If you get hold of an Excel file, you can open the Excel file and save it as a tab delimited text format or comma separated file format, and then you can use it for your input. So again, to check how big this data frame is, we can use the function that says not dimension, but that says shape. It says shape, but how do we call that specific function? We call that specific function by typing the name of the data frame that we want to call, and then mentioning a dot, and then followed by shape of operator. And if you type this, it's going to throw you an error because you should not. Yeah. So you give only shape, not shape of operator, only shape, and it's going to give you how big your this is, how big your rows, how many rows you have, and how wide your data frame is. That is what you are going to get. And what else do we have here? Sorry. You can also use something like data type. So let's see what this specific code does to our data frame. You can copy this and then execute it in the next Colab. Okay, it actually gives you something like the type of different columns that you have in your data frame. So it identifies each and every column to be this data type. This will be very, very helpful in the case if you have lots and lots of columns, and if any one of those columns does not have a starting number as starting value or first value as number. That's what we saw, right? When we saw the first value is not a number, the whole column is considered a character. So we don't want such a problem for our data frame, because we want to perform statistical operations and statistical functions, statistical analysis to our data frame. So we don't want that. So this is one of the way where you can check. A similar function also exists in R, which is a structure function. If you are interested, I can go there and show you how this function works in R. Let's compare that. In R, it's the data types function is actually given by structure of df2. Then if I run this, it's going to, okay, not structure, some str, maybe, yes, str of this one. So it's going to give you what each and every column is, right? It says a data frame of 15 observations, 52 variables, and it has so many different columns, a number, and all of these are number, etc. But it is mentioned as float in Python's case. So that is how we are going to refer, or that's how we are going to find out how what are the different types of data types of columns that we have. So this is one way where we can identify if any one of these data types or columns are not properly read. So so let's see, what do we have next in this data? Let's try this ourselves in the try it yourself column. If you are following me, let's open this in this lesson. I think I shared this lesson, because this lesson or try it yourself section in Python is organized little bit differently because of the console than it is in R. So in Python, you first have to run, or you first have to import the packages. Yeah, first have to import the packages, and this is how it is. Again, we have deleted some portions of syntax so that you can practice importing these packages. If you run as it is going to throw you an error saying unknown indentation error, unknown line. So in Python, it is very important for indentation. Okay, we can worry about indentation later, but now I'm going to type complete the syntax. I run this, success running code. Why is the answer incorrect? Maybe I have not cleared this sample. Okay, that's why. Maybe run now. Yes, success in running code. The answer is correct. So I have successfully imported pandas and numpy as pdn. So now in Python, in order to load up, or in order to take a file and load it into Python's console, what you have to do is it's the simple copy and paste the link is not sufficient. We have to download this file, like save link as, and then download it into your specific folder, and then drop the file into this specific part so that the console can actually read the file. So I have already downloaded this biological height dataset .txt, which is given here, and then I'm going to take that and where did it go? Yes, here. Okay. Let's close this and open a new one so that it's easy for us to navigate, make it small. There's no way to hide this or what? Hide routing meeting controls. Yes, that's what I want. Okay. So I wanted, where is this? I wanted this one to be in somewhere where I can access. Yes, sessions and demo. Okay. So this is the data set that we are dealing with. Let's take it and put it in, drag it. It's not letting me do it. Yeah. Drag it and drop a file here. And it is, yes, it is loaded. And then let's see, some portions of this sample is, some portions of this is not complete. You can see that by running, and it is going to throw us some error, most likely. Why it has not done it? And maybe it's taking some time to run. But basically, I can see that the separator is not complete and the header is not okay. Why is this? Let's clear the console. Let's run it one more time. Okay, something's wrong with this console for me. At least, did any one of you got this working in this console? There's something wrong with me. Let's refresh this console for me. No, you did not try. Maybe you can try it in Colab. Yeah, for some reasons it's not working. It should work. It's still loading up for me. Okay. Yes, go down. So in the try it yourself section, let's upload the files one more time. My internet is really slow today. Or my computer is taking a long time to load. Yeah, it's not. Wait, it's being unresponsive. Yes, okay, got there. Go up, go up, go up. Come on. Okay, let me stop my share and start my share one more time. Maybe it will work. Yeah, until then, yeah, I couldn't use it. It doesn't respond. Until then, can you please, maybe many of us are using it for the same time. That's why the console is not responding. If you have any other queries, please let me know until I get this working. Next, we are going to deal with how to subset or index the how to index the data frame that we have read in. So that we will definitely do. Let's see. Okay, we will deal with that one, and then maybe we can come back to this lesson. It's still not responding to me. Okay, now I can share my screen again. Maybe this time it will respond. Okay, can you see my screen? Okay, let's try import NumPy again one more time. Run this. This console worked. Yeah, I think many of us are accessing this for the same time. That's why it's actually freezing. It's not working. Yes, question, please. Go ahead, Emmanuel. Yeah, okay, let's abort this. And if you try it away from here, it will work. But basically, this is what you should do. You should actually download the file. Okay, wait it. If I execute this code in my workspace, I get an error. What is the error that you are getting? Can you paste the error that you are getting? Can check. So we will go to your Colab. Okay, we will go to the Colab. It's not okay. It actually ran, finished here. It actually ran successfully. Let's try it one more time in this Dropbox and this drop the file thing. Because you have not imported pandas as PD. So import pandas as PD and then do this, then it will work. Let's do this, and if I run, it's going to throw me an error. Yeah, first you have to import pandas as PD, only then it will work. Okay, this is not working. Let's go to you have, then you might not have imported. You might not have run it right here in the Colab. Are you using Colab? You have to execute this. Yeah, you have to execute this, right? You have to execute this, only then it will work. Like this, execute this, only then that will work. Only then it will work. So, okay, let's, VS Code, then it should work. It should basically work. Okay, so did it work there? So it did not give you any error while you executed this? If it gave you some error, then it might not have worked. Then if you have executed this, it does not give you any error. This rather work. Yeah, because no, but that is NumPy, right? NumPy work. You are generating from text file, that's fine, but this should work. Read underscore table from PD should work. If you copied and pasted, try typing it again one more time. Maybe there is some mistake. Maybe there is some font issue. Try typing it instead of copying and pasting. Okay, let's quickly. We saw how different types can be accessed. And as I mentioned while dealing with R, head can also work here. So here we have loaded everything into DF. So DF, first few lines can be accessed using head function. just like R, but just the calling is different in Python. You have to use head of data frame. Here you're using data frame dot head. That will work. And then just like how I described how we can index different samples in R and sample types and different columns and different rows in R and Python. In Python, as I mentioned, you have to